I'm finishing the biology V2 one tomorrow.
Okay.
Um, and conclusion is I'm actually very happy the test is now biochem because honestly the biology has been kind of like messing with me decent amount. So I wanted to go over some of the biology stuff and I feel like the passages are definitely different than what I'd see today, but it's still good review, especially of like I don't know, the organ systems and how to analyze passages still. So
Mhm.
yeah. Um, also, I
Mhm.
know today was supposed to be 6 to 7, but are you able to do until 8 by any chance or?
I have someone right after.
Oh, okay.
Yeah.
How about on Thursday?
Thursday, same. I have someone at like after.
And Saturday?
Saturday, same. Same. Basically, like,
That's wild.
because this week I'm uh taking off for 2 days, and next week I'm taking off for 2 days as well. So pretty pretty booked. Um
Okay. Wait,
yeah.
what days are you taking up next week?
Oh, it's gonna be Tuesday and Wednesday also for New Year's Eve and New Year's Day.
Okay.
Hmm. But after that, I'm pretty free again.
On Thursday next week, like, are you available? I mean, no, Saturday. Are you available?
Which Saturday?
So January 2nd and January 4th, like,
Hmm.
are you available past 7? Like so like 6 to 8, for example, is a possibility?
Yeah, we can do that if you want.
Okay.
For January 4th.
Um, January, yeah.
Okay.
Well, honestly,
Uh-huh.
no, for January 2nd. For January 2nd.
Oh,
Yeah.
you want to do a from 6 to 8, you're saying?
Yes, 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 yes.
Okay. All right. Updated it.
Okay.
Mhm.
So it's lagging. Okay. All right. So I can start. Uh, The ciliate protozoan Tetrahymena contains two nuclei, a diploid germline micronucleus and a 45-ploid macronucleus that is the site of gene expression during the vegetative state. Sexual reproduction in the Tetrahymena occurs by the process of conjugation.
This looks like an awful like figure.
Yep. Yeah. Um,
So it seems like little circle is a micronucleus, the square is a macronucleus, cell one, cell pair formation seems to be both of these cells pairing up. Step two, meiosis of the micronucleus and destruction of three of the four meiotic haploid nuclei. That's kind of like a polar body. Like it's not the same thing as a polar body because we're dealing with a protozoan here. Um, but it's similar to that. Um, Okay, and then mitosis, step three, mitosis of the surviving meiotic nucleus and genetic exchange. So the surviving micronucleus um, seems to have this genetic exchange between the pairs. And step four, fertilization of meiotic nuclei. And okay, and then so I guess we can see like the circle is like half white, half black. Um, okay. Step five, two mitotic divisions of the fertilization nucleus. So we have, another, or rather two more, or sorry, one divides into two, and then another one divides into two. Okay. Step six, differentiation of anterior nuclei into new macronuclei, elimination of some micronuclear DNA sequences, fragmentation of original micronuclear chromosomes into subchromosomal linear DNA molecules, two, differentiation of posterior nuclei into micronuclei. Um, so anterior nuclei turn into macro, and posterior nuclei turn into micro, apparently. Step seven, destruction of old macronucleus and one new micronucleus and separation of mating cells. Step eight, the first cell division yields four progeny cells. Each daughter cell receives one new macronucleus and a copy of the new micronucleus. Okay. And then after conjugation, the four sexually produced progeny divide by binary fission. Each daughter cell receives an exact copy of the micronuclear DNA and an uneven but approximately equal amount of DNA from the amitotic macronucleus. To minimize fluctuations in DNA content, small macronuclei undergo an additional S phase before division. That's when you double the DNA content. Um, and large macronuclei, oh, and the large macronuclei eliminate an S phase. So that's just to ensure that they have the same DNA content. Okay. Um, in the macronucleus, the genes for ribosomal RNA are located extrachromosomally. This suggests that the rRNA genes are, what are your thoughts?
Yeah, I mean, originally I thought that the answer was nonlinear because I don't know, in the nucleus they are linear, and I don't know, that's just what I thought. But um I mean, I eliminated B for sure, but I really wasn't sure. I just put nonlinear. Um
So they're asking me about a protozoa, right?
um yeah.
And the protozoa, so linear um would be something that sorry, the so like eukaryotes have linear RNA, right? Whereas like a prokaryote would have like could
Circular.
have like yeah, circular.
Hmm.
Um, so a protozoa, is that, how can you tell if that is a, a prokaryote or a eukaryote?
By looking at their DNA, maybe.
Um, so what's the difference between a eukaryote and a prokaryote?
The eukaryotes, they have the membrane round organelles,
And give me an example.
mitochondria,
How about another one?
nucleus.
Okay. So this protozoan, does it have a nucleus?
Yep.
So it must be eukaryotic,
Okay.
which must mean that it would have a linear uh, RNA, right?
Okay.
So A
Got
would
it.
be out.
I know the answer for this one,
Okay.
so can't like properly reason through it. But now, I mean, let's say to eliminate D, because the answer is self-replicating.
Uh-huh.
Now to eliminate rearranged, um,
Well,
I mean,
mm-hmm.
extra chromosomally.
Mm-hmm.
What would that have to do with them being rearranged? If it's intra-chromosomal or out extra?
Mm-hmm. Yeah. There's nothing, I don't really see anything about um that, but if these genes are not found on chromosomes and chromosomes are, you know, chromosomes are things that are, you know, replicated.
Hmm.
Um, and we know from the passage that or rather the question stem, that the ribosomal RNA are extra chromosomal, meaning that they are not on chromosomes. How could they then, you know, replicate if they're not part of chromosomes? Uh, they would have to be able to replicate on their own.
What does it mean for something to replicate? Sorry, like for RNA to replicate.
Um, just essentially making more of itself. Typically we see it in the context of DNA replication, and I guess this is talking about like ribosomal RNA.
Oh
Uh,
yeah, okay. Because you need more if you want more ribosomes.
mhm.
Yeah, okay.
Yeah. So, um, yeah. So like the ribosomal RNA, you need to make ribosomes, which you need to, translate proteins. Sorry, translate, like messenger RNA into proteins.
Hmm.
So we need our ribosomal RNA um to work basically. So it wouldn't make sense for them to be non-functional. They should have a function. They're not going to be non-linear because we know that they're similar to, or they're like eukaryotes because they have nuclei. So that means A is out, B is out. Rearranged, I initially thought of that possibly being like, when there's like whatever crossing over and genetic exchange, like in step 3 here. But I don't think I don't think that rearranged is the right way to put it. If it undergoes, homologous like recombination or whatever. But yeah, I think the main point for this question is that if you have some type of, you know, the gene. Oh, you know what? By the way, this is saying the genes for ribosomal RNA, not ribosomal RNA itself. The
Hmm.
genes for it, which means DNA, right?
Hmm.
And DNA, we know, uh, undergoes replication to make more DNA. So if the DNA for the rRNA are located outside of the chromosome, uh, that must mean that the replication of these genes, the DNA replication of these genes, uh, would have their own replicating mechanism that's distinct and separate from the chromosomal replication that happens during the S phase.
Okay.
Hmm.
An extra S phase occurs during amitotic division in a small macronucleus to minimize fluctuations in DNA content.
Okay.
This is most likely triggered by the presence of
So what does the S phase do?
duplicates the DNA content.
Yep. Causes so like that's DNA replication, right?
Yeah.
And I think they said something like, yeah, to minimize fluctuations in DNA content. So it's like, if you want to have the same amount of DNA content and an extra S phase can make it so that there's less fluctuations in DNA content, it's likely that this is triggered when you have a small amount of DNA. Because if you have a small amount of DNA and you want to minimize the fluctuations, that way, the extra S phase will make it so that you have a higher concentration of DNA.
Mhm.
So, uh, and since they're saying that this is in the macronucleus, then it should be low concentrations of DNA in the macronucleus, right? Right. The
Yeah.
centromeres could be something that functions like during the G1 phase or something, or yeah, the G1 phase where you make more like proteins that are involved in mitosis or meiosis, but the S phase is when you replicate DNA. So that would be triggered when you have low concentrations, I think. Yeah. Hmm. So like G1 and G2, you see how that causes like the more, well, I guess they're just writing chromosomes, but you're also making like cellular proteins and stuff that help the cytoskeleton, do its thing.
Got it. Macronuclei have the asexual progeny and Tetrahymena in the cytoplasm of the ova-producing cells
Oh yes,
of,
exactly
yep,
what I was thinking when I saw the
yep.
polar bodies or whatever.
Hmm.
So what would that be?
Share a common feature in that A, undergo uneven division, B, contain uneven amounts of nuclear material, C, regulate contents by adding or skipping S phase, D, apportioned at mitosis. So I know C is not correct. So I'm thinking A.
Hmm.
They both have the same amount of nuclear because I was between A and B.
Hmm.
And why did this polar body thing happen again?
Uh, just because, so in the egg cell, um, the egg is supposed to have an excess of cytoplasm and cellular contents because, like, from the sperm cell, you have just that nucleus, which is present in the head of the sperm cell. And just that nucleus at the head of the sperm cell, enters into the egg to during fertilization, right?
Yeah.
But everything else, like the cytoplasm, the mitochondria, and all of that other stuff, comes from the female, from the egg cell.
Yep.
So that means that the meiotic division of sperm cells would be, four, like when you have the two replications, or sorry, two divisions, you end up with four sperm cells because they don't really have much cytoplasm and you can evenly kind of distribute that. But for the egg cell, you want the egg to have a lot of cytoplasm and a lot of cytoplasmic organelles and things like that. So that's why for the meiosis of egg cells, you're going to get one egg cell and three other polar bodies, which are basically like small cells with small amount of cytoplasm that we don't want. We want because when you have a fertilization event, yeah, you're only taking the nucleus from the sperm, but you're taking everything else from the egg. And that means you want that enough cytoplasm for like a full diploid cell. So that's why, yeah, that's why for, yeah. Yeah, so here it says most of the daughter cells destined oh sorry. The when a vertebrate oocyte divides in meiosis, the cytoplasm is distributed to the two daughter cells. Most of it goes to the daughter cell destined to be the ovum. The other daughter cells, the polar bodies, are cast off with little cytoplasm. So you want the egg cell to have a ton of cytoplasm because it's not getting it from the sperm.
Yeah.
Hmm?
Okay. When an initially heterozygous macronucleus undergoes repeated binary fission, the result will be. So binary fission is asexual reproduction.
Okay.
And so you're just like self-replicating. It's kind of like mitosis.
Okay.
So to heterozygous macronucleus, the result will be loss of macronuclear chromosomes. Um, I feel like it wouldn't lose it. I mean, this might be referring maybe to.
Yeah, let me see the top. There. Okay. Two nuclei, a diploid germline micronucleus, and a 45 ploid macronucleus, which is the site of gene expression during the vegetative state. 45 ploid. So like haploid is one pair, diploid is two pairs. 45 ploid, I guess that means that there's 45 what copies? 45 chromosomes, I guess. Um, okay. Do they talk about binary fission on in the passage? Maybe on the bottom. Okay. After conjugation, the four sexually produced progeny divide by binary fission. Each daughter cell receives an exact copy of the micronuclear DNA and an uneven but approximately equal amount of DNA from the amitotic macronucleus. Okay. And, you know, okay. So initially heterozygous macronucleus undergoes repeated binary fission. The result will be loss of macronuclear chromosomes, increased rate of crossing over, production of macronucleus with a genetic origin distinct from the micronucleus, a variable allele distribution in the macronucleus.
Okay, I can eliminate B because crossing over is like meiosis, and I feel like there wouldn't be crossing over.
Okay.
Um, so ACD now. I feel like it wouldn't be C. I mean, it says each daughter cell receives an exact copy of
Hmm.
micronuclear DNA and uneven but approximately equal.
Hmm.
So like between A, C, and D, I'd probably eliminate C.
Okay.
Um, to be honest, I don't really like any of these answers, but like, I mean, one of them has to be correct.
Hmm.
So like, I just don't understand. A, loss of macronuclear chromosomes. Like, what? Why would it lose chromosomes if it goes binary fission? And D, variable allele distribution. Like, If it's replicating the same stuff over and over, how would it be variable? So I have no idea.
Okay. Uneven but approximately equal amount of DNA from the amitotic macronucleus. I think the word uneven can help us here.
C? Well, it would have to be C, right?
What up? What's up?
I would think it's C then.
Oh well,
It's
uneven, right? So C is just saying that there's an origin that that macronuclei have a different origin from micronuclei. But if we have an uneven, amount of DNA that happens during this binary fission, that could lead to a variable allele distribution.
why?
Because if you're having an uneven amount, so like, after so like binary fission happens and it causes each daughter cell to have an exact copy of the micronuclear DNA and an uneven amount of DNA from the amitotic macronucleus. So I see that as the macronucleus having, I mean, if it's an uneven amount of DNA from the macronucleus, if we look at our choices, A just says loss of macronuclear chromosomes. Um, B is increasing the rate of crossing over. C is talking about macronucleus and micronucleus having different origins. D says the variable allele distribution. That's the only one that seems to be matching what they said about the by after during or sorry during binary fission that you have an uneven amount of DNA from the macronucleus. So if you have an uneven amount of DNA, you'll probably have, you know, an uneven or variable distribution of some allele in the macronucleus.
Okay.
The amitotic division of the macronucleus will result in an uneven distribution of chromosomes, hence an unpredictable genome. Um, Given the large numbers of identical chromosomes in the macronucleus, however, repeated divisions are unlikely to result in the loss of a chromosome, so choice A is incorrect. There's no crossing over, so B is incorrect. C is incorrect because all macronuclei trace their origin ultimately to their micronucleus. I think they said that with anterior nuclei turn into new macro. Um, Fragmentation of original nuclear chromosomes into subchromosomal linear DNA molecules. Um, so I think that's telling us that the macronuclei come from the micronuclei. Yeah.
Okay. Some of the DNA sequences that are eliminated during macronuclear differentiation may be sequences involved in Figure 1, Step 6.
So I see step 6, differentiation of anterior nuclei into new macronuclei, elimination of some micronuclear DNA sequences, fragmentation of original micronuclear chromosomes into subchromosomal linear DNA molecules, differentiation of posterior nuclei into new micronuclei. Some of the DNA sequences that are eliminated during macronuclear differentiation, maybe sequences involved in,
Like, I mean, I would guess either A or C, probably A.
see, transcription, translation. Well, the only thing, okay, so translation should be out because that's RNA into protein, and they're asking us about DNA. So you can't really have a DNA sequence involved in translation, right?
Yeah.
It could be involved in transcription. It could be involved in meiosis. Um, can't be involved in ribosome production. Um, well, I guess that question that asks us about genes for ribosomal RNA. So, okay, maybe it could be involved in that, but I'm seeing that as more something with ribosomal RNA. So I'm thinking of either transcription or meiosis. But if we're eliminating DNA sequences, and we know that, so before that, so let's see, step 3, okay, step 1, cell pair, step 2, meiosis of the micro and destruction of the of 3 of the 4 meiotic haploid nuclei. Step 3, mitosis. Step 4, fertilization of meiotic nuclei. Step 5, more mitotic divisions. Macronuclear. Mm, what are your thoughts?
I mean, I would just choose transcription and move on, but yeah. I have no idea whether this is meiosis or mitosis. First, it says there's two mitotic
Yeah.
divisions before, then it says meiotic. I don't know. Like this, I just hated this passage, honestly. I'd rather read 5 blocks of text and look at this artwork.
Yeah, this looks not fun. I mean, so I think they said something about
Um, I wanted to try to get one more passage in before 7. So
okay.
I think, okay, cool. It's C. What? We're supposed to know what the macronucleus is? Like, on the old MCAT, I guess, or what? Uh,
So I guess they're saying that the macronucleus, its purpose is to carry out transcription and translation, which requires ribosomes as well, and the only one that's different is meiosis. because transcription, translation, and ribosome production all have to do with that central dogma. But yeah.
In the mating of two tetrahymena strains that are homozygous in their macronuclei and heterozygous in their micronuclei for a recessive gene, what percentage of the F1 generation will express the recessive phenotype?
So I think the main thing that we want to see is the difference. What's the difference between a macronucleus and a micronucleus? So if you scroll up on the top left, they say that this macronucleus is the site of gene expression. So that can be where like the macronucleus can is probably when you have where you have, DNA transcribed into RNA, RNA translated into proteins. Um, and let's scroll down on the left. So they're saying that this maybe the micronucleus is the one that undergoes the meiosis, like the um yeah. So I think that I think that means that the macronuclei are responsible for gene expression and micronuclei are responsible for like the sexual reproduction.
Mhm.
So so we would look at the fact that the micronuclei here is heterozygous for a recessive gene, and that'll give you that 21 25 percent that have the recessive phenotype. I think that's the main thing that we have to take away from this passage. Okay, yeah, here we go. So micronucleus for reproduction, macronucleus gene expression.
Mhm.
So yeah.
Wait one second.
Are you going to see family for like tomorrow or New Year?
So basically, my family's in New York,
Yeah.
but my older brother's fiance lives in Miami.
Okay.
She's from Miami. And
Mhm.
my older brother's visiting, like her, here.
Mhm.
So I'm like getting dinner with them tomorrow.
Cool. Nice.
Like, cause she's Cuban and like I don't know if you've heard of Noche Buena. It's like really big in Cuban culture. I personally haven't heard of it till like last week, so but it's like their Christmas Eve and it's like an insane thing. Um so I'm gonna go there, meet like her whole family and stuff, but yeah.
Noche buena.
Yep.
Good night. Cool.
Okay. Let's try to get through this as much as we can.
Yeah, it snowed here like on the first day of winter, like a week.
Yeah. I saw on social media some people's like like pictures of people were taking
Mhm.
outside or whatever, and I saw snow, and I'm like, oh wow.
Yep, yep.
Um,
Pretty cold. It's like 27 degrees right now.
yeah, that's crazy.
Hmm.
I won't tell you how much it is here because then you're gonna want to kill me. Okay, let's do this. So E. coli, a bacterial, or do you want to read this? So, or should I? Because we only have 20 minutes.
Um, you could read it.
Okay. E. coli, a bacterial species that inhabits the colon, benefits humans by breaking down foods for which we lack digestive enzymes and by producing vitamins such as B12 and K. About every 20 minutes, E. coli undergo asexual reproduction. Copies of the bacterial chromosome, which are attached to the cell membrane, move to opposite ends of the cell as the cell divides. Plasmids contain various non-essential genes, including genes that confer resistance to specific antibiotics. Because plasmids are not attached to the cell membrane, they may not be equally distributed among daughter cells. Um, Bacteria also carry out a form of sexual reproduction called conjugation. Conjugation is when plasmid genes from one bacterium direct the building of the cytoplasmic bridge or sex pilus between the organism and the bacterium lacking such genes. Following plasmid DNA replication, a copy of the plasmid DNA moves unidirectionally through the pilus. Is it pilus or pilus? What do
Hmm.
you say?
Either or.
Okay. Bestowing new genes upon the recipient cell. Conjugation may occur between members of the same or different bacterial species. A man was hospitalized with a ruptured appendix and received massive doses of the antibiotics ampicillin and kanamycin over a period of weeks. Shortly after leaving the hospital, he contracted M. tuberculosis, pathogen that caused tuberculosis from a coworker. He was again hospitalized and treated with large doses of first ampicillin and kanamycin with no effect. Recovery was rapid when a third antibiotic, streptomycin, was administered.
Yeah, so they'll probably ask about, you know, the some of the pathogen that survived after those antibiotics were given, and they probably pass that on through the sex pilus,
Mhm.
something like that. Okay. The bacterium containing genes for sex pilus construction gave rise to a daughter cell lacking these genes. This most probably occurred because
So I I didn't think it was D. It said one copy of the plasmid containing genes was digested by a bacterial lysosome. They say one copy, which means there could be other copies, which means those might still exist. I don't know. I didn't think it was D. And then I was between A, B, and C. I had no idea how to eliminate them.
all right, let me see. So um copies of the bacterial chromosome which are attached to the cell membrane move to opposite ends. Plasmids contain non-essential genes like those that confer resistance. And they are not attached to the cell membrane. They may not be equally distributed among daughter cells. Okay, so maybe we're looking for something that has to do with them not being equally distributed, right? So, um,
Yeah, I put C. I don't know if it's right or wrong, but
um,
because that's what
mhm,
that talks about distribution.
mhm. Yeah, could be it.
Okay, it is. Nice.
Mhm,
Okay. Now, next.
mhm.
At the end of his initial hospital stay, a few E. coli cells remained in the patient's colon, even though he was taking antibiotics. These cells were most likely present because.
Mhm.
So, my reasoning was the E. coli cells developed some sort of drug resistance.
So when we talk about that, right? if you think about it in terms of evolution, um, it's not like the antibiotics created some drug resistant mutation. It's more so that through like everything in evolution is really just random mutations.
Hmm.
And so some E. coli would have random mutations that would let them be resistant to some type of antibiotic. Uh, right? And
Okay.
ones with those random mutations that let them be resistant to antibiotics just happen to survive when uh you know um take antibiotics, and it would just kill everything else, but not the ones that have those random mutations. And so the ones that have those random mutations that make them resistant to the antibiotic, they can just make more of themselves.
Got it. Great explanation. Okay. Then there would be D.
Yeah.
Okay. I didn't actually know that. I thought that I mean, that makes sense. But I thought that they literally develop the resistance.
Yeah. That would be like more like Lamarck, the guy who was like, giraffes have long necks because the tree
Yeah. Okay.
they have to stretch, but that's
Yeah.
right. It's really just random mutations, and the environmental factors make it so that some of them die and some of them don't die, but it's never like, resist your mutations will be random. Yeah. Hmm.
Okay. Tests performed on the M. tuberculosis strain infecting the patient's coworker indicated that the strain was susceptible to both ampicillin and kanamycin, and the coworker was successfully treated. The M. tuberculosis most likely survived in the patient because it had.
So they said um that the um okay, that the plasmids can contain the genes that confer resistance, and when the plasmid gets, you know, passed on, it's done through conjugation, aka through the sex pilus, right?
Okay.
So that's what we want to look for.
But my question is, why can't it be B?
Adapted to its new metabolism by modifying
B,
adapted to
B.
its new environment by modifying its
B, 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 B. B as
oh.
in banana.
Undergone an well, it's not going to be, you're never going to have that. You're never going to have like an antibiotic induced mutation. You're going to have random mutations that just make it so that some uh
Got it.
just happen to be resistant to the antibiotic.
Okay. Now I know. Now I know. Yeah. Okay. But yeah, B, A. I mean, D and C
Hmm.
don't really make sense. It was between A and B.
He
Um,
says it reproduce more rapidly. Yeah.
okay. The patient's ruptured appendix required treatment with antibiotics because he had a bacterial infection caused by, so A, and tuberculosis. Um, yeah, well, that's not the answer because
Yeah.
he after he,
Huh?
hmm?
Oh yeah. What were you
It
gonna say?
happened after he left the hospital.
Yeah. So I would look at either B or C because that's the only one that says from the appendix, right?
Yeah.
Because what happened here is because he had a ruptured appendix. So if a ruptured appendix causes some bacterial infection, that bacteria has to come from that appendix, right?
Okay, right.
So,
So now I also saw that
Mhm. And then it's between colon or the abdominal cavity. So if an appendix ruptures, it would just kind of go out of that of the appendix, which is attached to the colon. So if you have a ruptured appendix, it should enter into the general region rather than something that's connected to it.
right.
It's like if you had a ruptured small intestine, it wouldn't make sense for the thing to enter into your large intestine, right?
Hmm.
Because if it wasn't ruptured, then it would make sense because that's the next place that it goes. So my guess here would be E. coli and yeah. Mhm.
Okay, so when it's yeah,
You wrote the appendix is continuous with the colon so that bacteria can move between these two structures, but a ruptured appendix would allow the E. coli to enter into the abdominal cavity, which is not normal. Yep.
following the antibiotic treatment, the patient's colon will be recognized by other bacteria. The factor that will ultimately determine whether antibiotic resistant E. coli will persist in the colon. I this was A. I got this right, I think.
Hmm.
None of the other answers just made any sense. Like,
Yep.
maybe B, but like not really.
Hmm.
Um,
Yep.
so for this, like, um, do I have to know this? So ask about organelles.
Which of the following organelles most resembles a Golgi apparatus when an intact eukaryotic cell is viewed under the electron microscope? Um, well, the Golgi, like, what does it look like? It's kind of like sacs, membranous sacs, right?
I guess. I don't really remember what it looks like, but yeah.
If you look at, you know,
Just search up. Yeah, yeah.
yeah, you would see that it's like, well, the endoplasmic reticulum is like these sacs,
Yeah. Hmm.
um, and the endoplasmic reticulum would also be like that in those sacs, whereas a
Hmm.
nucleolus is, I guess, like not in like a sac, but kind of like some area where like whatever, like ribosomal RNA and stuff can be concentrated, and the mitochondrion is you know a dual membrane organelle, plasma membrane, dual membrane as well. But yeah, we're looking for like membranous sacs, like SACS, and the closest thing should be, when I say that, I mean like, it's like folded and stuff,
Yeah.
like this kind of,
No, no, I know. I found pictures.
yeah.
Yeah, okay. That makes sense. Um, oh, this one. So a resident of famine area appears undernourished and extremely emaciated, has eaten only starches for the past 3 months. We cannot digest starches, right? Or can we? What is the one thing we can't digest? There's starch, cellulose, glycogen.
Cellulose,
Ah, okay.
because it has beta glycosidic bonds. Um, okay. Eaten only starches for the past 3 months. Urine analysis shows that a large amount of nitrogen is being excreted. Where is that going to come from?
Nitrogen,
Yep.
proteins.
Yep.
So it was between B and D. Um, like how would you eliminate the other?
So D is saying incomplete reabsorption of nitrogen products due to kidney failure. If that's the case, then that just means that, well, okay. If that was the case, you would have perhaps more um of that, I guess, urea being excreted. But the person being like malnourished and stuff would point me in the direction of the person's muscle is being broken down. Um, so that would make more sense, I would
My
say.
other question is, so why does like, you know how sometimes there's those questions about like you're breaking down beta fatty acids through oxidation, you have the ketone bodies in your urine or whatever. Um, and then there's also like here, it's like there's nitrogen in the urine because you're breaking down proteins. Why would it end up in the urine? If we're breaking it down, are we gonna absorb it? Like we're gonna use it? Why
Uh,
would it be in the urine? Why would we excrete it?
what was your question again?
So you know how there's like sometimes when you're breaking down a lot of fatty acids, right? There's the ketone bodies that appear in the urine.
Huh?
Same here. You're breaking down proteins. You're breaking down proteins and nitrogens are in the urine. Why are they in the urine? Why are we excreting this if we're breaking it down to use it?
Um, so I guess if we're breaking it down to use it, I think we would be using it for let's see, for energy. So I'm thinking of the urea cycle, and you can break the proteins into like like if you have like a glucogenic amino acid, a ketogenic amino acid, it can be broken down into like some type of like thing that can contribute to producing sugar. But I think like acetyl-CoA is where they all what they all of the breakdown of whether it's fats, proteins, sugars, like they're all gonna have acetyl-CoA. Sorry. Um yeah,
Uh,
acetyl-CoA is the common thing.
yeah, but why is it in the urine is my question.
Hmm. So I think that just that they'll all like you can use up the amino acids that you break down. And so hold on, let me look up something. The urea cycle. Okay, so the urea cycle involves proteins being broken down into things that could be used for energy, but you're still going to have urea as your waste product. So basically, amino acids can be broken down into ammonia, and that's like a toxic chemical. And so it can if you convert the ammonia into urea, that can be a non-toxic thing that you can excrete out. So I think that's what we're doing.
Okay.
Yeah. I would take a closer look at the
I
urea,
will. Hmm.
but like my cat, so like cats are obligate carnivores, like they only eat meat. So their urine has a ton of ammonia. Like, I don't know if you have experience with cats or anything like that, but yeah, they because they only eat meat, um, their urine has like a ton of ammonia. But yeah, my thought is that when you're breaking down proteins,